Hello, everyone. I'm Haley Kopczynski,
And I'm Rizwan Roy.
and we are talking about the composition and function of carbohydrates within a cell. So the composition for carbohydrates consists of carbon, hydrogen, and oxygen. Typically, the imperial structure is represented by CH2O. You may recall back to an organic molecule lesson in your biology class, and you might have discussed the monomer for carbohydrates, which are monosaccharides, also known as simple sugars. So monosaccharides can be known as polyhydroxyl aldehyde or polyhydroxyl ketones. So this is whether it has a hydroxyl group coming off of the carbon chains. The picture to the right shows the three different monosaccharide sugars, glucose, fructose, and galactose. So that's in the picture here to the right, so you can see those, and then they have the structure. So these structures of the carbohydrates can be drawn or represented in three different forms. So you probably have seen the open chain form, which I have a picture of on the right here, and I'll talk about these different structures, but those are the long straight chains. And then you have the acetyl structure, which is in the form of those ring structures that we've seen on the last slide. The first carbon will be put with the OH group on the fifth carbon to make the ring. The last way you'll see carbohydrates represented is in the Haworth structure, which is when it is in the presence of a pyranose ring. So the picture to the right shows that open chain structure, and for our activity, we'll be working with the ring structures. There are different types of carbohydrates, the first being the simple sugars. These are monosaccharides, mono meaning one, the saccharide meaning sweet. The most common of the monosaccharides is glucose, and the number of carbons that are in monosaccharides range from 3 to 7. All sugars can be identified because they end in ose. When a simple sugar has an aldehyde group, it means that you'll be able to see a CHO structure known as aldose. And if it has a ketone group, this means that in the structure, it's going to be, it'll have both a C and O double bonded, and this is known as a ketose. So the picture on our last slide, you can see the aldose, so the CHO, and then the ketose, which has that double bonded C and O just there. So again, you can have up to 7 sugars. So you may also see your tris, your pentose, and your hexose, your triose, which are 3, and which were also shown in the picture on the last slide. So you have your triose, pentose, and hexose sugars. Disaccharides are whether there are 2 monosaccharides together. When this occurs, there is a dehydration reaction that occurs. Therefore, one of the hydroxyl groups on the monosaccharide combines with a hydrogen from another monosaccharide, and this releases water to form a bond, more specifically a covalent bond. And this bond is known as a glycosidic bond or linkage. There can be alpha or beta bonds of this type depending on where the group of rings, or where the plane group is on the ring plane. And the last type of carbohydrate that we'll talk about is the polysaccharides, and this means many sugars, so it will be a long chain linked together by those glycosidic bonds. Okay, so let's take a look at some of these different types of carbohydrates more specifically. What kind of sugars fall under monosaccharides, disaccharides, etc. So when we look at this picture, I really love it because it breaks it down a little bit further. So in our monosaccharide group, these are simple sugars, and they cannot be further broken down through hydrolysis. And it's going to be our glucose sugar, which is the most common form. And then we have our fructose and galactose. Then we have our disaccharides, which again, these are the 2 sugar molecules put together. So we have sucrose, lactose, and maltose. And then in the picture, we also have the oligosaccharides, which range from 2 to 10 sugar molecules. And we have our glycosidic linkage that we discussed. And the two examples are raffinose and ribinose. And lastly, we have our polysaccharides, which have more than 10 monosaccharides in them. Each polysaccharide differs from each other depending on the type of chain links and linkage and the type of branching that they have off of them. And their function, again, is to provide energy, and they also store energy in our bodies, which we'll talk about a little bit later. When you hear that, it's going to be, you'll hear starches and so examples are starches, glycogen, and cellulose. So you'll hear that word glycogen, hopefully again, because that's what we store in our body for energy. All right. To demonstrate learning about different structures of carbohydrates, we'll do a bead activity, and this activity can be done in small groups with 2 or 3 individuals in each group. Things that you'll need for this activity is different colored beads. So for the activity, I have put together white beads, orange beads, and yellow beads are going to be used. And you can use any color beads as long as you have 80 beads for one color, 10 and 10 for the 2 other different colors. Students can section off the different colored beads and then label the plates. So you need to get some plates so they can put the beads on. That way they're not rolling all over your floor. Some scissors, and then you'll also need to get some different colored pipe cleaner. So 3 different colors of pipe cleaner. Ziploc bags are also really nice to put the material in, and then you can just hand out that to the students to make it a little bit easier. So again, I chose 80 white beads for the glucose, 10 beads for the galactose, and 10 beads for the fructose. In the second picture I have here is the example of how some of these will look when putting together. So for example, if I had students put together 2 glucose molecules making a disaccharide, that's going to be the 2 white beads on the pipe cleaner there. Or if they're putting a maltose sugar together with a galactose sugar with a glucose sugar. And then I also have a couple of pictures of what the polysaccharides would look like. So these can be done together as a starting point with your students as a teacher-led activity, and then you can break away and have students going into a little bit more in depth. So here's what a worksheet would look like to kind of give them, and there's more to it to kind of break things down as they're going through, and you can have them ask them different questions to ask them to build different structures. So you would ask them, what's the difference between these 3 groups? And they would say, well, it's the color. So that they know that there's a difference between these different sugars. But all of them are going to be the same size. And then they can go into looking at the number of carbon atoms. So they can look at these different structures and get used to working, like counting the different oxygens, the different hydrogens, and working with the structure of these different sugars, which is really neat. And then it also shows them like how many orange beads would be on these. So again, when students have a pretty good grasp of what they're doing, as well as the students have a good idea of single bonds, simple molecule structures, and understand that there's dehydration reactions occurring and hydrolysis, they can start working with their groups and having them fill out this worksheet. And they can bring out their bags and examine what's in them. And again, you can choose to work through this with your students, or you can have them do it on their own and then do a group review at the end. So that's our activity there.
Okay, and now we're going to talk about the main functions of carbohydrates within the cell. So there are 4 that are basically recognized, or within the body actually. It's for energy production, energy storage, to build macromolecules, and then for sparing for proteins and fats. So I'm going to get into a little bit about each of those. So for energy production, glucose is the preferred source of energy within most cells. And what happens is cells that require energy from they remove glucose from the blood with a transport protein within their membranes. And the way that they get the energy from glucose is the bonds between the carbon atoms and how they're broken down. And what happened was, as you know, in photosynthesis, sunlight energy is used to produce those bonds in the process of photosynthesis. And then we take in glucose from those from the plants that produce them by photosynthesis. And then we undergo cellular respiration in order to break down those bonds through a process called glycolysis. And our liver actually ends up redistributing 70% of the digested glucose to other parts of the body through the blood. Let's see. And so, yeah, cellular respiration basically ends up being a controlled burning of glucose versus an uncontrolled one. And a cell ends up using many chemical reactions and multiple enzymatic steps to slow the release of this energy so it's not an explosion, and to more efficiently capture it, so that to capture the energy that's held within the chemical bonds in the glucose. Now, as for storage, what happens is, if your body already has enough energy to support all its functions, that excess glucose is going to be stored as glycogen. And most of that is stored in the liver and muscle tissues. The liver, like muscle, can store glucose energy as glycogen, but in contrast to the muscle one, it's going to sacrifice its stored glucose energy to other tissues when it's low. So that's so if your blood glucose gets low, it's going to pull from your liver the glucose that's there and use it, or the liver will send it to other places to be used. Let's see. Approximately 1/4 of the total body glycogen content is in your liver, which if you're looking that in terms of time energy, you're looking at about a 4-hour supply of glucose. But that is dependent on the activity level. So obviously the higher the activity level, the less that time will be. And the liver is going to use the glycogen reserve that it has as a way to keep the blood glucose levels within a narrow range between meal times. So that's what helps sustain your energy between when you're eating. And when the liver's glycogen supply is exhausted, glucose is then made from amino acids that they get from breaking down proteins in order to, and all of this is tied into homeostasis. Okay, so now we're going to talk about the other two functions. Which, all right, when we're building macromolecules, what happens in this case is that some glucose is converted to deoxyribose and ribose, which is important. They're the sugars that make up your DNA or RNA. In addition to that, we also have that, it's also used to make NADPH, which is important for protecting against oxidative stress. And it's also used in all kinds of chemical reactions throughout the body. And if all of the energy or glycogen storing capacity and building needs of the body are met, then that glucose is going to be used to make fat. So this is where the issue of if you're taking in too many carbohydrates and not utilizing them enough throughout all your bodily functions, this is where you start to gain weight. And so what we mean by sparing proteins and fats means that is if you have an adequate glucose supply, it ends up not needing to break down proteins to get amino acids so that it could make glucose or getting energy. And so what can happen is that, like we said, glucose can be synthesized from amino acids, but that you only want that to happen in like emergency cases. You want to make sure that you're taking in enough glucose so that it's not breaking down your proteins, because that's what happens if your body needs to make glucose from your amino acids, it's at the expense of your proteins. And so keeping adequate glucose levels will avoid that. And And the reason why it ends up breaking down the proteins is because we don't have a way to store amino acids. They're used. So there's no storage of them, which means they have to, in order to get amino acids to make the glucose, it has to break down the proteins. And most of that comes from your muscle tissue. So that's where it also can get kind of dangerous for you if that's what's going on. And as in terms of sparing fats, as your blood glucose levels rise, the use of lipids as an energy source is then inhibited. So what happens is glucose additionally has a fat sparing effect there. And that's because an increase in blood glucose stimulates the release of insulin, which is then telling your cells to use glucose instead of lipids to make energy. Okay, so that's all about the function. So now we're going to talk about the activity that we'll do to demonstrate so that you can see how carbohydrates are used within bananas, or how their presence is, and so that this can make it clearer in terms of how carbohydrates are inside of living things. So what will happen in these activities is there's this activity actually has 4 levels to it, but we're only going to have the students do 2 of them related, because we need the tissue level and the cellular level 1 in order for them to understand what we would like for them to understand about this. So it involves learning about carbohydrates at different biological levels. So the first level is at the tissue level. And what students will do there is they're going to sample ripe and a not ripe banana. And that will lead to a discussion about what they observed about the difference in taste and other things like this, texture and all that. And then from there, each group is going to then have a ripe and a not ripe banana of their own that they're going to do a starch test on, which using iodine. And then after that testing is done, students are going to record their data and discuss the process of hydrolysis. And by the end of that discussion, hopefully they're going to reach the conclusion that starch gets broken down into sugar, which is what made the ripe banana taste sweet and the not ripe banana not as sweet. And from there, you can also pair that iodine test with testing of purified starch and sugar so that they can see the results as well. So they can just get a better understanding and more a bigger visual. Then from there, they're going to go to activity number 2, which is exploring them at the cellular level. And what happens there, or in this activity, is students are going to discover where in the banana the starch is found. And so they're going to observe the iodine tested bananas through a microscope. They will also observe, and based on that, they're going to get to see different starch levels within the bananas. And so what they're going to do is prepare slides with the banana that tested positive for starch, as well as the non-ripe banana. And they're going to observe them all under different powers in the microscope, because you get to see different things in the banana cells when you do observe them under different powers. After that, students are going to record and draw what they observe. And from there, it will lead back to the discussion, hopefully, once again, tying it into the fact that the ripe banana has the starch broken down into sugars, and then the non-ripe banana, there's still a heavier amount of starch in there. And hopefully by the end of all this, they'll understand, the process that's going on, that starches get broken down into sugars, which is then used to power processes within the cell. And that is all of our activities. Here are our references. And that concludes our presentation. Thank you.
Thank you for tuning in.